Okay, so it's my great pleasure to introduce Casey Huang. Dr. Huang is a professor of bioengineering, microbiology, and immunology at Stanford University. Casey did his PhD in physics at MIT with Joanne Polis. I might not be pronouncing that correctly. Did I just butcher the name? Joanopoulos. And then undergraduate degree in physics and math at Caltech. So the Huang Lab focuses on understanding the biophysical processes of bacterial cell growth and cell size, both at the molecular and population levels. And it's pretty amazing to see Casey, Dr. Huang's lab span so many different scales, ranging from all the fundamental biophysics, a molecular scale to the microbial community scale. And so that's a really amazing set of systems to capture using a variety of different techniques and modeling and experiment. And Casey has won many awards for his research, including the NSF Early Career Award, also the Science News Top 10 Scientists to Watch in 2017. And we're really excited that you were able to give a virtual seminar. We were hoping to meet Dr. Huang in person, but we're fortunate to have you join us virtually and give your seminar. We're very excited to hear your talk. And with that, I will turn it over to Dr. Huang. Thank you so much.
Thanks, Ophelia, and thanks everyone for their understanding. I also was looking forward to the opportunity to see everyone in person, and I hope that everyone is safe and sound. Let me just turn on my, let's see the laser pointer.
Yes.
Great. So it's really a pleasure to be speaking with all of you. I know this is a quite diverse department, and I, as Ophelia mentioned, come from quite a different background. So if there's any questions during the talk, please just unmute and ask away. And I'm happy to divert the talk in any way that helps understanding. I thought I'd just give a brief introduction to myself. This was me right before I started my lab in 2008. I had far less gray hair, and I was thinking the blissful thoughts of a theorist. I was a theoretical physicist who fell in love with bacteria, basically thinking about questions about structure and spatial organization, as Ophelia mentioned. This is a David Goodsell painting. And I looked at E. coli cells and I thought, well, there's all these fundamental questions about how they determine their shape and size, how do they determine where to divide, what sets the spatial organization of the molecules inside. And really, this ended up driving the first 5 or 6 years of my lab, where from a physicist point of view, my goal was to build reductionist bottom-up models using tools like single molecule imaging and molecular dynamics. But also, I quickly realized that the sort of wonder and complexity of biology is that one has to really understand the phenomenology at the same time. And so we used things like quantitative imaging with microfluidic perturbations to characterize cell growth. And the question was, how do you connect these bottom-up and top-down methods? And we found that computational modeling, which was part of my background, was a really critical tool in the case of cell shape and cell size. The cell wall was kind of the right intermediary, and we developed models to predict the shape and dynamics of the cell wall during growth. And that's not what I'm going to talk about at all today, but I thought I would just show this, this cartoon that summarizes that first period of my lab. This was made by Jorge Cham, where he tried to capture some of the things that we think about, the fact that there are many shapes and sizes, and how we have to bridge these scales, and both how we use genetic perturbations to try to adjust cell size and ask about the fitness of different mutants, but also mechanical perturbations like osmotic shock. And ultimately, the goal was to understand what are the rules that go into bacterial cell growth. And I think for me, a lot changed when I taught a class with my good friend Justin Sonnenberg. He's a glycobiologist who specializes in how things like diet affect the gut microbiota. And he and I taught a class that was meant to teach biologists about computational modeling and data science. And through the course of this class, we kind of realized that there was a similar paradigm that we were hoping to address, where we could take individual subunits, in this case cells, and try to understand the phenomenology at the tissue and organismal level. But that part of the class really ended up focusing on new tools and computational and quantitative analyses that we could use and bring to bear on the gut microbiota. Hi, Doug. So over the course of today, I thought I would tell you about 3 vignettes that capture how we're approaching this problem. The first was actually at the host scale to try to understand the robustness of the gut microbiota to antibiotics and pathogen invasion. And then I'll tell you about some work we've been doing to develop synthetic communities and also to understand the statistical properties of the gut microbiota. And hopefully both those will be make more sense in the coming slides. First, I just wanted to set the stage by telling you about both it was intellectually interesting, but also frustrating project that was carried out by Kat Ng in the lab. And she was a fantastic postdoc who started with the question of how predictable is the response of the gut microbiota to antibiotics. And so, kind of the challenge that we started with was, well, if you start to read the hundreds, if not thousands of papers on antibiotics in the gut microbiota, you can end up with very contradictory findings. So this paper from Wolf-Dietrich Hart's lab said that there was a massive restructuring of the microbiota due to streptomycin, and where you see this tenfold drop, if not more, in load. Whereas this paper from Brett Finlay's lab showed that there was basically no effect of streptomycin. And I'm not arguing that either one of these is wrong. In fact, I'm sure they're both right in their own way, but certainly for someone outside the field, this was very confusing to try to wrap your mind around. And so we just started with a very simple list of what we thought were known knowns, and then what would we do to try to understand this? So one thing is that antibiotics are used to kill bacteria. We can all agree on that, and that they have very different mechanisms of action, and hence they kill differentially in vitro. We also know that bacteria develop resistance, which is a huge issue, and it's become increasingly appreciated that hosts are more susceptible to infection by pathogens during the recovery after antibiotics. And so what Kat did was she said, okay, well, how are we going to hope to come up with any sort of defining principles or findings? So she said, okay, first, let's take a mouse with a certain gut microbiota. We apply an antibiotic, and then we can start to ask, okay, well, is there a small effect? Is there a large effect? Is the effect differential across taxa? And then how does the microbiota respond or recover after that? Also, how does it respond to then, or how is recovery dependent on the presence of either the same microbes in the environment or different microbes? And how does this respond to host dependent factors like fiber? You could have a very fiber-rich diet, or you could eat double doubles all the time. So the experimental design for these next few experiments I'm going to show you is very simple. She takes germ-free mice that have been gavaged with human feces. So these are so-called humanized mice that have a human-like microbiota. They've been equilibrated and they're treated with antibiotics for 5 days to mimic a human treatment. And then we sample at various time points to capture the pre-antibiotic state, the disturbance, the post-antibiotics, and then the recovery phase. And I'm just going to show you some data from 2 antibiotics. We did this across 7 different antibiotics, streptomycin and ciprofloxacin. And one of the first things that she noted was that if you were to simply sample the total bacterial load once a day from streptomycin treatment, basically you would conclude that nothing happened. And so what I mean by that is that all of these blue points are high, except for this point. But I want you to note that this was actually a fecal sample that she took after 12 hours, quite rare in a mouse experiment. But she did this because we were concerned about potentially fast dynamics. And it's quite remarkable that there's a 5 order of magnitude drop after 12 hours that then really rapidly recovers over the next 12 hours. The same thing roughly happens with ciprofloxacin. It's a little bit longer time course, but I just want to stress that even during the antibiotic time course, you see recovery. This could come from any number of things that I'll get into in a second. But again, you see this several order of magnitude drop and then recovery 1 or 2 days later. And this is mimicked by the compositional dynamics. So you don't need to pay too much attention to the different classes, but the main point here is that the yellow bars, which are representing the Bacteroides, decrease over the course of the first day. But again, they recover during the antibiotic treatment, which is over these first 5 days. And the same is true for the rest of the taxa. So I think it's interesting because, of course, it says that there's a certain inbuilt robustness even during continued treatment. But also, it was something we sought to ask, well, how much of this is something we can easily understand based on basic principles? And so one possibility is that all of the Bacteroides strains became resistant to streptomycin, and that's how they recovered. And so what we did was we isolated many Bacteroides strains pre-antibiotics. We actually found out quite interestingly that they were all resistant already, which makes it all the more bizarre that they take this massive decrease in relative abundance, compounded by the fact that the total abundance of bacteria has dropped by 5 orders of magnitude. So this is one of the intrinsic puzzles about in vivo antibiotic resistance or sensitivity, that in this case, all the Bacteroides were already resistant and yet they transiently died. So just to introduce a couple more puzzling or interesting aspects about in vivo biology, one of the factors that we also wanted to explore was diet and how does that affect the antibiotic response. So here I'm showing you that diet lacking polysaccharide, complex polysaccharides causes this really prolonged loss of both bacterial load and diversity. So diversity drops by about 90%. And you can see here now, there's no recovery at all during the antibiotic treatment, which is this period in purple. The last thing I just wanted to throw out there is, well, what happens if we remove the environmental reservoir? So here now what Kat did was to take a cohort of mice and separate them into singly housed and co-housed mice, and then compare the response to streptomycin, since that was the one where we saw the largest effect on bacterial load. And one thing we found was that there were dramatically different community dynamics. So in the co-housed mice, we saw the same thing that I showed you before, where the Bacteroides drop and then recover over the period of the antibiotic treatment. In the singly housed mice, what you see is that the Bacteroides don't recover at all during the antibiotic treatment. But even more strikingly, if we look across individual mice, the individually singly housed mice actually show completely different responses. Every mouse looks different from the other. These are 2 mice. We saw a completely unique response in all 6 mice. And so that suggests that the ability to reseed from an external reservoir may be really important for understanding robustness to antibiotics. So putting it all together, I think there's part of this which suggests that sensitivity may be a community phenotype, that diet can induce a long delay in recovery and reshape the community response, and that absence of reservoir can disrupt the community response. But I think the thing I really want to emphasize about all of this is that these 3 points were part of a really painstaking 5-year process of many mouse experiments. And even then, there's so many parts of parameter space that you may be thinking to yourself that we should have studied instead. And you're exactly right. For instance, there's nothing in here about how the dose affects the community response, even though I'd love to do that experiment. And so I think the question we were left with here is, how do we start to deconstruct these complex systems? And so this is where a student of mine, Andres, picked up. And so he referred to his thesis as the ecology of poop teas. That's somewhat in jest, although poop tea is actually a concept that goes back to, I think, the 4th century BC, where it was actually used as a therapeutic in Chinese times. Probably a better title is this one, where we sought to develop communities that could be used in vitro to study the gut microbiota. And to be clear, the idea is to model the gut microbiota, not necessarily the gut itself. In fact, part of the goal is to highlight which parts are microbiota and which parts might be gut. So I'm going to tell you about a top-down approach to building in vitro communities. And the idea was to take humanized mice and ask, could we derive stable and complex communities from these fecal samples? And of course, the answer would be no. In fact, the first time I proposed this to a student, they said, this is the dumbest idea ever. Like, the only thing that's going to happen is that E. coli is going to grow and you're going to sequence E. coli like 1,000 times. I have to say, this is one of the rare times that I was actually right. But the fact that we were able to get complex communities actually means that we have the potential also to study isolates and then go from bottom up to build communities from those isolates and then use them to recolonize mice. And so I'll show you parts of every arrow along this way over the course of the next several slides. The first thing we did was to actually make use of the fact that we had this ongoing experiment where diet and antibiotics could be used to change the gut microbiota. So what we did was we took these humanized mice, we took 2 mice from each of the 2 diets, and then sampled them at these 4 time points, days 0, 1, 5, and 14. And as I showed you before, the composition is both diet dependent and time dependent, but is pretty constant across co-housed mice. So I'm showing you here the standard diet. Here's the polysaccharide deficient diet. So you can see here, we thought this would be a great starting point because I have essentially 8 different starting points from which to derive communities. So starting with all of those 16 fecal samples, what we did was to inoculate them into 4 different media. These media are quite complex. They're selected based on a pilot experiment. And then we passage them over 7 passages where every day they get inoculated into fresh media. And they're kept at 37 degrees the whole time. They're kept anaerobic the whole time. So basically they're kept in an environment that is as close to what we can do to mimic the human gut. And this ends up amounting to 192 communities that are being passaged every 2 days. We did this either for 7 passages or up to 16 in some cases. And in each case, what we did was to try and characterize it using tools like phase microscopy, using flow cytometry, and things like 16S sequencing. And that's what I'll show you the bulk of. I want to stress that they're kept anaerobic and at high temperature the whole time. And so to sort of allay the concern that all you're going to get out is E. coli, this is actually one of the first communities that we passaged. And this is after it's reached a stable fixed point. And obviously from the cell morphology, it's very clear that there's a huge amount of diversity, different shapes and sizes, even these absolutely massive organisms. I don't know what they are, but they're certainly pretty bizarre. So what actually happens? Well, quite satisfyingly, not only does the community stabilize very quickly, but it actually stabilizes to a composition that is fairly reminiscent of the starting point. So the fecal inoculum is shown here in the left-hand bar, and then all the passages are shown here on the right. Basically, it's the same from passage 2 onward. And if you just compare the relative abundance of each family in vivo versus in vitro, there are families that drop out. Those tend to be ones that are below 1% in abundance. In fact, the only major change was that the verruca microbia, which are shown here, are actually switched out for proteobacteria or Enterobacteriaceae. That's not too surprising because the verruca microbia actually like to eat mucus. We don't have any mucus in our media formulation, but everything else remains at relatively the same composition. So we were excited about this. We said, okay, this is a reasonable starting point. They're also highly replicable. Essentially, these are 3 technical replicates. They basically look the same. And not only that, if you look at the species level or the ASV level, basically all 3 communities are virtually the same. So the last part, which we found very satisfying, was that remember that we have different starting points. So we took different fecal samples from mice at different points during the antibiotic treatment. And so what you can see here in the light colored circles are all of the initial starting points, the fecal inoculum. And the main point here is that the communities that we derive from each one of those different starting points do not end up at the same point. In fact, they end up at a point which is very close to where they started. So you can see here the pink overlap with the pink, the brown with the brown, the green with the green. In fact, the only ones that show a little bit more dispersion are the blue, the peak, and we suspect that's only because much of what we sequenced during in the fecal inoculum was actually dead cells because they had just been exposed to the antibiotic. So, in fact, not only does this show that it's both that the inoculum can have a large effect, but actually it's also the case that medium can have a large effect. So this is in another medium called TYG, and it looks roughly the same as BHI, but other media like GAM and YCFA actually look quite different from the initial inoculum. So it doesn't mean that bugs care what they eat, which is not so surprising. I will say that one of the parts that was quite surprising to us is that YCFA is a medium that was designed to culture a large number of gut microbes, and it actually fails miserably to maintain community composition. It also fails to maintain diversity, whereas BHI and to a certain extent TYG can actually maintain a very large fraction of the initial species, in some cases up to 80 or 90%. And so this was comforting that we also weren't losing a lot of the species that we started with. Last, just to give you some data about the efficacy or the utility of these communities, we took one of the communities and put it back into mice. And what you can see here is that the original community composition that we started with and what we ended up with after deriving the communities and then putting them back into mice is actually very close. In fact, the only thing that shows a large change is, again, the verruca microbia, because it was lost during passaging. Not only that, we took the fecal samples after we colonized these mice with the community, and we did host proteomics. And here, I just want to emphasize that if we compare the humanized versus the community colonized mice, we see a very strong correlation, in particular with the immune-related proteins, whereas this correlation is partially lost when we compare with germ-free mice, either the humanized or the community colonized mice. So this suggests that the community is going back in, establishing roughly the same composition, and reprogramming the host proteome in the way that you would expect from a native microbiota. So what can we actually use these communities for? One question was, can we actually understand anything about our antibiotic experiments that we did in vivo simply using the communities? And so what Andres did was first just to expose the community to repeated ciprofloxacin treatment. And so what he sees is that indeed the Cipro has an increasingly large effect. If you compare communities derived from stool samples pre-antibiotics versus ones right after antibiotics, you can see that the residual communities actually have increased resistance. That's not so surprising. In fact, what you can see here is that there's a progressive decrease in the yield of the community, of the pre-antibiotic community, as you add more and more ciprofloxacin, suggesting that you're eliminating more and more species. Whereas in the case of the residual community, it actually maintains its yield throughout, suggesting there's some robustness to the antibiotic treatment. So what happens to the communities themselves? Well, what we see is now we can make use of the fact that the communities are amenable to things like dose-dependent experiments, which would be very difficult to do in mice, not only because of the cost, but also because we have no way to actually regulate how much drug gets to the intestines. What you can see here is that some of the families go extinct, as you would expect, and some of them are maintained. We can study this across different doses, and you can see that certain families are particularly resistant to the antibiotic treatment, whereas others seem to show a dose response. In fact, what we can do is to isolate organisms from this community and then ask how much can we predict simply by knowing the individual species responses to antibiotics? And the answer is some, but not all. In particular, you can see a positive correlation here between the MIC and the fractional change. So the species that are more resistant actually end up being overrepresented in the community after antibiotic treatment, and vice versa for the sensitive species. But there is dispersion, and I think that dispersion comes from interesting effects like competition for resources, and I'll get into that in the later part of my talk. So, however, the experiments I just showed you don't really mimic the experiments we did in mice. What we did in mice was to treat with ciprofloxacin and then release a ciprofloxacin. And so Andreas did the same thing in vitro. What he did was to treat once with cipro and then passage in every way you can imagine, with different without cipro, with cipro, without cipro, and then without cipro, and then back with cipro. So what you can see here is that once you release the ciprofloxacin treatment, there is some degree of recovery of the community, but not completely. And so this jives with what we saw in vivo, that there is a long-term disturbance to the microbiota that occurs through antibiotic treatment. We can also see, though, that if you allow the community to recover, you can actually see that species or families that were undetectable during the antibiotic treatment actually end up recovering, like the Erysipelotricaceae and the Enterococcasiae. And so it suggests that these families are actually resilient to the antibiotic treatment. They disappear, but then they come back. And we know that it's resilience and not resistance because we can treat with the antibiotic again and they go away. And so what this allows us to do then is to start comparing what happened in vitro to what we saw in vivo. So remember, in vivo, we saw certain families were able to persist and then recover. And what we find if we compare these two experiments is that not only can we recapitulate all of the different survival characteristics that we saw in vivo, but in vitro, we can start to tease apart how much of that was because of resistance, how much of that was because of recovery, and how much of that is dose dependent. And I think this is a really critical aspect because, of course, what we want to understand is what's the sensitivity of the gut microbiota to resistance, and what can we count on for recovery, in some sense, getting for free without, for instance, having probiotic supplementation. So just in a last part here, I just want to emphasize that we can actually dig even further here. And in fact, one of the families that was particularly interesting was the Bacteroideaceae. The Bacteroideaceae actually ended up having a large amount of recovery. And we wanted to ask how much of that was due to resistance and resilience in and of itself. And so in vivo, what we see is that if we explore the Bacteroideaceae, there's one member, Bacteroides vulgatus, that manages to recover very quickly. And then in the post-recovery period, there are several Bacteroides that recover, but not all of them. There are about 14 that were present to start with. So for instance, Bacteroides ovatus is one that doesn't recover at all. And so what we can see is that continuous treatment of our communities ends up selecting for a subset. You can see here that at low doses, B theta survives, at intermediate doses, CAC-A and uniformis survive, and at large doses, vulgatus survives. And already you can see here that vulgatus doesn't survive at intermediate concentrations because of competition against these other two Bacteroides, presumably. If we then look, so this I would characterize as some degree of resistance depending on the concentration, but also this transient treatment experiment where we allow 2 passages of recovery allows us to see things like Fragilis recovering at low doses, Infantis recovering at high doses. Actually, excuse me, at intermediate doses, we don't see any recovery at high doses. And so this suggests that some of these species then have inbuilt resilience. If we then look at this characterization, we see that this actually matches, precisely with what we saw in vivo, where vulgatus clearly has resistance, where there are several other strains that show resistance at other doses, but that the recovered strains like Fragilis and Intestinalis are the ones that we see only coming back after we relieve the antibiotic pressure, and things like ovatus end up extinct no matter what you do. So this was satisfying for us because this set of antibiotic experiments took place over the course of a week, as opposed to the much longer timescales of the in vivo experiments. I just wanted to show you one more example of a potential use case of these communities. So I mentioned at the beginning that antibiotics sensitize hosts to pathogen invasion, and one very well-studied example is Salmonella. So we know that many mice strains are resistant to colonization, but then antibiotics will sensitize will sensitize mice to salmonella. And so what we did was to derive communities from pre-antibiotic and post-antibiotic, just like I showed you before, and then introduce salmonella at different doses and ask whether it's able to survive after 48 hours of culturing. And so with this in vitro salmonella challenge, what we found was that the pre-community essentially could get rid of practically all of the typhimurium. This was even with a 1-to-1 dose of salmonella in community, whereas the other communities were essentially susceptible to complete salmonella invasion. This is just plate counting, which is not particularly quantitative, but illustrates the qualitative aspect. This is where we actually count salmonella cells. And there's just this massive change where the pre-community essentially has gotten rid of the salmonella. So I think this has both an interesting discovery potential, but also as a potential diagnostic where the communities themselves seem to retain a lot of the colonization resistance phenotypes that we attribute to the gut microbiota itself. So, I think in terms of obtaining communities that are complex and stable and reproducible, I didn't show you, but they're also very storable and scalable. But I think the fact that they can mimic composition to some degree in the gut, but even more so mimic the dynamics of perturbations is really what has us the most excited about pursuing this in the future. I think, as I'm about to show you, we can also start to get at some general ecological principles and also use these in the context of mice, use them to understand antibiotic resistance and colonization resistance. And so I think this set the stage for Andres' second project, which was to try to use these communities to understand assembly and resource utilization. So this was work done with Alvaro Sanchez's lab at Yale and his postdoc Sylvia Strela. And this was, what we did was then we took 8 human donors and derived communities directly from their feces, and then explored growth across a very large number of carbon sources spanning most of central carbon metabolism in E. coli. So all the ones in colored bold writing are the ones we've explored. So this is 23 carbon sources, and there's kind of a general punchline about what happens to the community. So first of all, if you look with unweighted unifrac, so what this means is it's basically judging who is there, not how much of each species is there. They basically all segregate by donor. So each one of these colors is a different donor. You can see that by carbon source is just a big hairball. However, if you use weighted unifrac, which now incorporates the abundance of each organism, you can see here that now by donor, it's a hairball, whereas there's pretty good separation across carbon sources. And so this title sort of says it all. The donors say who's there, the nutrients dictate their abundance. And you can see here that there's actually an interesting correlation between where they sit in principal component space and the growth of the community, the OD of the community. That's what's colored the dots. So what's actually in these communities? Well, interestingly, if you look across all carbon sources and then average across all donors, you see this range of yields or biomasses of the community. And there are certain families like Enterobacteriaceae and Enterococcaceae. These are fast-growing species where their absolute abundance, and basically we measure absolute abundance by simply multiplying the relative abundance by the OD. You can see that there's a strong correlation between how much the community grows and how much of these families there is. By contrast, there are a large number of families that exhibit roughly constant abundance. And this is constant down even for very low levels, like the desulfuribrios are present at 10 to the minus 3, 10 to the minus 4, same with the porphyromonaceae. And so we found this really interesting. This basically suggests that all of these, for instance, the Bacteroides, are ignoring the fact that the community is growing much more. I'll get back to what this means in a second. So what Andres did was a neat set of experiments where he grew a community for 48 hours. And every hour or 2 hours, he would take an aliquot of the community. And he would sequence that aliquot, calculate their relative abundance, but then use the OD of the passage culture to calculate the abundance of each family at each time point. So what I'm showing you here is effectively a growth curve of every family throughout a passage cycle. And what you can see here is that those fast growers I told you about before, the Enterobacteriaceae and Enterococcaceae, are the ones that rise the fastest. And then there's other timescales at which other community members will grow. You can see here we've broken it down for the Bacteroideaceae and the Lactobacillus, and these are ones that have experienced a sort of a secondary wave of growth. These two are the fastest growing species amongst these families. So our goal was to try to see if we could understand what was going on here and what the consequences were. And I forgot to say one thing, which is that in every one of these experiments, what we did was we supplemented a community with we supplemented a complex medium called BHI at low concentration with an additional carbon source at high concentration, where the carbon source was at fixed molarity across all carbon sources. So just to try and break this apart, what we did was we isolated 15 members that account for a large fraction of the abundance. They account for about 70% of the abundance of the parent community. They also cover most of the major families. And then you can see here the growth curves of each one of those isolates. Again, the Enterobacteriaceae and the Enterococcaceae are the fastest growing ones, and you can see here other time scales for the rest. This was, I think, a day when Andres got really bored with writing his thesis. And so this is actually the actual line of succession for the US president, except with some other names. And they're all specifically chosen because you can notice here that Escherichia and Enterococcus, Lactobacillus and Bacteroides, and so on. This is actually meant to presage one of the main findings that I'm about to show you about, which is that there is a predictable line of succession. And so the main punchline that I'm going to show you is that we can predict what happens to the community by understanding what happens to each individual species. First, I just want to emphasize that if we reconstitute those 15 members back together, they reconstitute with roughly the same abundance as in the parent community. So we think of this as a reasonable model for the full community, but it's one that we can manipulate. And I want to emphasize that E. fergusonii, which is the Enterobacteriaceae, actually occupies a relatively unique niche. What I mean by that is if you grow the 15-member community, it grows to basically the same yield as the full community. If you grow the 14-member community that is missing the E. fergusonii, you can see here that this difference between the 15 and 14 is basically the same amount that E. fergusonii grows by itself. And so that suggests that it's basically is filling its own unique niche, and that'll be important in a second. This is emphasized by the fact that what this is showing is that how much E. fergusonii grows in the community, starting from all these different dilutions. So we can actually dilute the culture of E. fergusonii by 10 to the minus 7, basically down to a single cell. It will still grow to the same number of cells in the full community. So I also want to, actually, I'm going to skip over this for now. So coming back to this question of how much can we predict from the individual organisms, what I'm showing you here is the growth of each of the 15 strains across the 23 carbon sources. And this water just means in BHI alone. So you can see here, many bugs can eat many carbon sources. Some are more fastidious than others, but many are generalists. So why is it that things are so predictable in the community, meaning that there are so many families that just don't seem to have any variation across carbon sources? And it's because growth occurs first on the complex medium. So what you can see here is that the first period of growth for all these strains looks the same, and then something else happens. And you can see here this for a very broad range of all of the species. And here, what I've quantified is the growth rate. So you can see here that the growth rate is basically the same until it starts to run out of the BHI, the first thing it's eating, and then it shows some carbon source dependent effect. Okay, this is an approximation, but it's a pretty good approximation across all of the species and all of the carbon sources. And the same thing happens in communities. So what I'm showing you here now is the absolute abundance of each species within the community, calculated the same way I told you about before, where we sample every hour. And so what you can see here is that everything looks the same at the beginning, and then things change depending on the carbon source. So again, it looks like you grow first on BHI, and then you grow on something else. So what that means is that we can calculate something like the excess growth for each community. So here, Sij is the fractional share of the strain I in carbon source J. And the way we calculate it is we basically just say, how much more did it grow with the carbon source? Did this strain grow with the carbon source relative to the overall yield of the community in that carbon source compared with BHI alone? So it's essentially like, how much of the carbon source did you get? And so we compute this for the parent community. And you can see here that in many cases, the E. fergusonii gets a lot of it. Sometimes the Enterococcus gets a lot of it. Galactitol is actually the only case where they don't take over. So now what we can do is we can say, well, how would we use the individual strain data to calculate what happened in the community? So imagine you have the growth curve of an individual strain. You could say, well, I can use its yield. I can use how quickly it grows, which I'm going to capture with the area under the curve. That actually, I'm going to tell you, works terribly, as you might expect, because so many of them are generalists. Instead, what we can do is we can say, let's identify a time, this TEQ, where now the total amount of excess biomass that has been produced in each of the isolate cultures adds up to the total yield of the community, right? So it's as if I was allowing them all to grow separately, but once they get to a collective yield that corresponds to them having consumed all of the nutrients, that's it. That's all you get to grow. And then I'm going to calculate the AUC for that time point. So this AUC star. What we found was that we could actually, I'm going to show you tests of this model using the 15-member community and then the 14-member community where you're lacking E. fergusonii to test what happens in this line of succession as you get rid of the species that is the strongest grower. And so basically the growth of the culture looks roughly the same, depending on whether you have a 15-member or 14-member community. In the 14-member community, there are some changes that that we'll get to in a second. But essentially, they have very similar architectures. Here is the parent community. Here's the 15-member community. They look qualitatively the same. And in the 14-member community, you've gotten rid of the best grower here. And so now the Enterococcus seems to have taken over. But in the 15 or the 14-member community, you see the same relatively constant abundance of families across all carbon sources. So I told you we wanted to ask whether we could predict what would happen in the community using the single strain data. So here is the data where we calculate the excess abundance, and here's now the prediction with final OD. It doesn't agree at all with the data. The same with the area under the curve. If we use the final OD at this special time point that we've inferred where everything would get used up, things still don't look great. But if we compute the area under the curve at that time point, now things look quite similar. And here I'm showing you a correlation between the data and the prediction, where I take the ratio of the Enterococcus and the E. fergusonii concentrations. And so these two are pretty strongly correlated. And so it suggests that there's enough information if we incorporate both when we think things will run out and how quickly the strains were able to grow in order to get to that point. It's basically just saying the early bird gets the worm, but we can quantify exactly how early you have to get there in order to win out. We can also predict the dominance in the 14 and 11-member communities, where now when we get rid of the E. fergusonii, the Enterococcus dominates. And again, the AUC does a poor job, but the AUC star at the special time point does a much better job. And the same, to a certain extent, is true in the 11-member community, where we get rid of the Enterobacteriaceae and all three Enterococcaceae members, where now we can again predict the winner of the competition, which ends up being Arachnospiraceae. So I'm going to skip over these slides to just these three slides to just emphasize that I think the punchlines of this part of the story are really simple. It suggests that certain species have very well-defined niches in a complex medium, like the Fergusonii, and that if you have this stratified growth where everyone goes for the complex medium first and then goes for the simple carbon sources, then excess growth is going to be dominated by the fastest members. You get there fastest, you get to eat it all, and that's great. But it's not just that one guy will win. You can actually predict the dominant members and maybe more in a complex community simply by understanding the growth of each species in isolation from a quantitative point of view. And I think it's exciting because I think we can start to then parse apart what parts we don't understand as well, because I think that will tell us about other interactions that are important. So in the last few minutes, I just want to tell you about a kind of the flip side to this bottom-up approach to understanding community dynamics. And that's looking from the top down at the statistics of gut communities. So this is work by a postdoc, Po-Yi Ho, and collaborator Ben Good, where we'd like to exploit the intrinsic statistics of gut microbiotas, and in particular link this to potential fluctuation in nutrients. So this is a time series from Lawrence David's lab, where they sampled the gut microbiota every day for about 4 months. And we know that despite the fact there's very complex interactions, the gut microbiota can remain remarkably stable over even years. And so as evidenced by the fact that this yellow species remains very high, the blue one is most of the time the second highest, and so on. But I also want to call your attention to all of these fluctuations. And you might say, well, okay, that's just noise, but that noise actually has very interesting character. So there was a paper from Dennis Vitkov's lab a couple years ago where they looked at the statistics of these time series and found something really interesting. In particular, what they see here is this this Laplace distribution, where the probability of seeing an abundance change with a certain log fold is actually linear in this log space. Okay, so this power law distribution is something you don't see in many cases. This is not a Gaussian. And it's linked to several other statistics that were interesting. So for instance, they linked the variance in each species to its average abundance. There was a linear correlation in log space. They also looked at the residence and return time. So this is how long does a species remain present and how long does it take to come back after it goes extinct? Because some species will go below the limit of detection and come back again. And then there are several other statistics that they presented. There was no particular underlying model that they were able to use to explain this, but it was interesting because they saw this in both mice and humans. So we wanted to ask what mechanisms could generate these statistics. And so we made use of what's called a consumer resource model. So essentially in a consumer resource model, you assume that there's a certain number of resources, M, and a certain number of consumers, N. And then there are some rates of consumption, R. So this matrix here tells you how quickly does each species eat each metabolite. And these differential equations are quite simple, and they just simply describe the dynamics of the system. One of the things that we added to this, which I think makes a lot of biological sense, but is also very important, it turns out, is sparsity. So we generated this matrix, and then with some probability, we set each element in this matrix to 0. And the probability at which you go to 0 is what sets your sparsity. And so I'll come back to that in the next few slides. So what Po-Yi does then is he now makes the assumption that the variation in species abundance is due solely to fluctuations in the resources. Basically what he's saying is that changes in your diet are what drive changes in your microbiota. And so he assumes that the initial resource amount on each day is fluctuating with some noise strength and some correlation. So that means that if you go away from your normal diet, you come back with some time scale or correlation strength K. So then what he does is he simulates the system over and over in each day to come to a macroecological steady state. And then he asks, now the next day, when each resource changes, how does that change the steady state abundance of each microbe? We have to make the assumption that there's some small reservoir of species that competes for resources, because otherwise things can only go extinct. They can't ever come back. But with a very small reservoir, then what he can do is to get abundance time series that look at least qualitatively like the up and down plots that I showed you on the last slide. But even more remarkably, actually, these look from a statistical point of view extremely similar. So for instance, we can ask how it compares to a human gut microbiota. And for instance, we can take our data and ask, how does the variation scale with the mean abundance? And in fact, we can find parameters that fit this data precisely. We can also ask how the distribution of abundance changes that I showed you can be fit to an exponential, just like I showed you before. It has this peak triangular shape. And in fact, as you might surmise, the width of this distribution is precisely dictated by the noise in the fluctuations in resources. We can also calculate the residence and return times and match those extremely well as well. And we can also calculate something that wasn't picked up on before, but which is for each species, we can calculate how much abundance change there is relative to its relative abundance. And that distribution of restoring slopes, right, how quickly does it come back to its normal relative abundance? We can actually match that quite well as well with the model. So across all these statistics, we were able to find one unifying model. So this is essentially an existence proof that it's possible to match the gut microbiota statistics with a single consumer resource model with fluctuating resources. But even more than that, what was interesting is we can ask, how do all the parameters in our model, of which there are not very many? There's the ratio of the number of species to the number of metabolites, the sparsity that I talked to you about S, the noise strength in the fluctuations, and the noise correlation K. And what you can see here is that if you measure several parameters in the statistics, for instance, the richness or the diversity of the community, the slope of how strongly variance is coupled to abundance, and the restoring slopes, and the width of the distribution, that basically each one of these readouts is tuned by one of these four variables. So each one of these four variables has a sort of physiological interpretation in terms of how it adjusts the macroscopic statistics of the community. And basically in all the gray boxes, the curve is relatively flat. So that allows us to zoom in on the parameters that specifically match this microbiota. But not only that, we can take these four parameters and these four readouts, and I'm going to skip this for now, and we can also look at a large number of microbiotas. So for instance, we can look at a different human microbiota and see the same matching of statistics, albeit with a different set of, a slightly different set of parameters. We can also look at a human saliva microbiota, where now the parameters change quite a lot. We can look at vaginal microbiotas. In fact, vaginal microbiotas show huge differences in the sense that some are very diverse and some are dominated by a single lactobacillus species. But in fact, the statistics of those communities look almost exactly the same. So it suggests that despite these large differences, there's kind of the same underlying model can explain the data. And so what all of this tells us is that we can look at all these different microbiotas and start to place them in the space of macroecological parameters that are relevant for describing their statistics. You know, for instance, we can start to ask how human and mouse microbiotas differ. And what we find is that human gut microbiotas are under really intense resource competition and have more families and effective resources, which means they have high niche overlap. Whereas mouse gut microbiotas are, in some sense, the reverse. The human saliva actually has, what you can see here in pink, seems to have very low sparsity, which means they have very few metabolically distinct microbes. And one of the things that we find is that the range of noise strength demarcates the typical fluctuations and perturbations that we see in environmental resources. So we think this is exciting because it basically allows you to now start understanding in a very small parameter space how all of these different microbiotas may be differing. And I think the fact that the time series statistics constrain these macroscopic parameters so strongly suggests that these parameters may be physiologically relevant numbers that describe the properties of the microbiota. I am going to now just jump forward to my acknowledgement slide. And so this is from our last lab retreat, which was now several years ago with Will Luddington and Fred Chang's lab. We have a really diverse set of people in the lab, from biochemists to bioengineers to physicists to biologists and geneticists. Anyone's interested in talking about future opportunities as postdocs or students, I'll be really excited to talk with you. I also want to just thank everyone in the lab and many collaborators and funding sources. And thanks for your attention. And thanks very much for sitting through another Zoom. I'm really sorry not to be there in person. Love to be looking at you in person, but hope to do that in the future. Thanks very much.
Thanks, Casey. That was a great talk. I think we have some time for questions. Maybe I'll just jump in quick, ask a question. So I was wondering, with the consumer resource model being able to recapitulate some of the statistics of the distributions of species abundance, that's pretty interesting. Did you have a null model of some kind to show that the consumer resource model is unique to be able to recapitulate that? Because yeah, I would imagine with like many
it's a great question.
Yeah, I mean, if you with enough parameters, you can fit. You can probably explain a lot of data, a lot of trends in the data.
4 parameters you can fit E. coli, and 5 you can fit elephant.
Right.
So yeah, so it's a great question. I have 2 thoughts about that. So one is, what are some reasonable null models? Well, one null model is complete lack of interaction, and so I skipped over it. But you can take a non-interacting null model that nevertheless has the same steady state, and ask whether you can predict species correlations. And the answer is not at all, unsurprisingly. But in fact, the consumer resource model, in perhaps a non-intuitive way, does predict the species correlations quite well. So the blue is the data, and the orange is the consumer resource model. We had not actually expected this to even be a statistic that you could predict. So that's one aspect, but that's possibly a straw man, because we know that there are interactions. So you might say, okay, well, what about a Locke-Volterra model that's based around pairwise interactions? And so Locke-Volterra models and consumer resource models are not necessarily different. You can consider them in some regimes to be mathematical representations of the same model. And so what we did was we took our consumer resource model with fluctuating resources, and at every time point, we generated a new Locke-Volterra model that was the same at steady state at each time point. Now, of course, it's a new model each time, but that's okay. Like, the consumer research model also has new inputs at every time point. So the answer is you can predict many of the statistics, which is not surprising, because of course, we knew that our starting point of the consumer resource model worked to explain the statistics. But I think the thing that's really interesting then is you have to ask, well, is that a particularly interesting ensemble of Locke-Volterra models or not? Or is it just that we found like that unique mathematical representation that works? And what we found was that if we generated 5 different instances of the consumer resource model by just having different noise in the resources, that the distribution of correlation coefficients between the Locke-Volterra interaction parameters was completely different in each case. So I think that's my best piece of evidence that Locke-Volterra models are probably not the right way to go, because every time you have to hit like this incredibly special part of parameter space, we think, to be able to explain the statistics. I think there's much more to be done along those lines, but that's the best we could do, because of course we can't exhaustively explore Locke-Volterra models because the parameter space is too high. I hope that makes sense.
Right. Yeah, I guess with the GLV model, actually, why don't we move on, because I could keep talking forever about GLV. Drew, do you want to jump in? You had a question?
Yeah, hey, thanks for the great talk, Casey. I have a question back at vignette number 2, I think, with the 15 and 14 member communities, and
Yeah.
this idea that in these simplified, complex communities, the early bird gets the worm, and everybody's using whatever's in BHI before they use whatever you've supplemented into the medium. But BHI has like lots of amino acids, lots of
Yeah.
glucose, and glucose is, candy for everybody. I wonder, if you were to have BHI without glucose, that could be one experiment. Would that model work? Or if you were to grow these bugs at like a steady state where you're constantly spilling in a little bit more medium, would you expect the outcomes to be different?
Yes, totally. In the latter part, totally. In the glucose part, no. And the reason I say that is because basically every species behaves the same way in glucose. They eat the BHI before they eat the glucose. In fact, I got to get to the bottom of this somehow. Like you are not the first, and I was definitely one of the people who would say, yeah, everyone loves to eat glucose. And basically that is only a coarse approximation of reality, as we have found, in the sense that same thing with MRS and a lot of the fly microbiota species, that we sort of were working under that assumption. In that case, we did exactly what you said, which is to take glucose out, and it didn't affect things at all. And so it really looks like they're staying away from the glucose for quite a while.
So they're eating the amino acids.
They must be, I think. Yeah, I mean that's the obvious candidate. We haven't actually done that experiment. I mean, it would be good to do, but we see the same, that everyone undergoes a second phase of growth once they get to the glucose. It's not affecting their growth in BHI.
Cool.
Yeah, but I think your point is well taken. If I can restate it, which is that there's definitely a way to break this rule. And I think that for me, that's the interesting part, that like, that like, we got to understand like, what are the scenarios in which one rule will apply and another one, and then how to transition away from that. Because I think we've had this like underlying question for a long time, which is, for instance, how is batch culturing different from chemostat-like environments? And there's so many different ways that it is the case. And I'm hoping this like gives us like an inroads in terms of specifying that more precisely.
Great. I think we have one question through the chat. Haley, I don't know if you want to ask your question, or I can just read it out loud, either way. Okay, how about I read it? Does a perturbation, antibiotic, stress, diet dictate how the community recovers, or do you see the same recovery progress despite type of perturbation?
Yeah, that's a great question. So the answer is yes and no. So there are there are definitely totally different responses that one can see to, for instance, different stresses than antibiotics. And in fact, we've done a screen of the in vitro communities across about 1,000 different antibiotics and drugs. And what's particularly interesting was to see how the community can move as you apply a perturbation. But I think the recovery process is one where we see a lot more convergence in terms of how the community will try to come back to its steady state. And I can't say anything too intelligent about it, but I'll tell you what I'm hoping to be able to find, which is that what we see in vivo is that if the community can recover, then there is a very stereotyped recovery trajectory. But when it can't, then things go haywire. And I'm hoping to see that in vitro, where the harder you hit it, the more you sort of like knock it into a space where it's not gonna come back into the same well of species composition. But I think more to be done to establish that. And I think there's a question from Dan as well.
Yes, do you want me to, I can read it. Have you looked into how the length of antibiotic treatment affects microbiota recovery in your in vitro model? Many clinical courses of antibiotics last weeks. Do extended treatment regimes do more harm to the microbiota?
Yeah, that's a great question. And I think there's a really interesting side note to that, which is that how much does the recovery process then depend on what one would assume is the continued buildup of resistance as you treat with antibiotics for longer and longer. We have not looked at that at all, but I think it's a fascinating question. I think the part where what I suspect is going to be the case is that you will start to see, assuming you do not go to a case where you completely abolish all of the any species which is not completely antibiotic resistant, I think you'll start to see really interesting hierarchies of resource competition in the same way that I showed you with the different doses, where you can see that at one dose, a very resistant guy will survive, whereas at a lower dose, two species can outcompete it. And so I'm hoping this will start to tell us a little bit more about who is actually competing with whom when you now look across time instead of dose to see the trajectory of recovery. But I think it's a great idea.
Okay, I think we might have time for maybe one more question, and then we'll have meeting questions offline. Any other additional questions for Casey?
I have a question. So you saw that a specific Bacteroides, its growth depended on both what its monospecies MIC was, as well as competition with other Bacteroides species. Do you have a sense of how frequent that is, where it doesn't just depend on its monospecies MIC, but also interactions via competition?
Yeah, it's quite prevalent, and I think the one of the things I can perhaps a systems level demonstration of that is I told you about the screen across 1,000 different drugs, and so I also told you that the Escherichia species in this community actually has a relatively isolated niche from all of the others. And so that's a case where what we see is that the correlation between its MIC and its sensitivity in the community is almost precisely correlated. And so that's kind of like it's kind of like a positive control that, if you're growing something by itself, then, growing it by itself in a community is the same as growing it by itself in isolation. Whereas what we see with many other species is that there's a large amount of dispersion, where overall there's a positive correlation between the MIC and its sensitivity in the community, but there are many drugs that are counterexamples and outliers to that. And I think those are precisely the ones where it's revealing the competition with other species that are modifying that relationship. And so I think there's much more to be done there, but we see that in basically every in vitro case, barring the Enterobacteriaceae, and in virtually every in vivo experiment as well, actually, that as soon as one thing takes over, in fact, even if it's highly resistant, even during the antibiotic treatment, anything that was able to stick around and outcompete it, in fact, starts to outcompete it and then eventually wins out when the antibiotic is taken away.
Cool, thank you.
All right, let's thank Casey for a great seminar.
All right, thank you so much.
Thank you so much. That was very interesting. All right.